Yeah, hello everyone. Good afternoon, good morning, or late good evening, wherever you are. This is the International Space Science Institute with its Game Changer seminar, where in this series of 10 talks that started in January, we're looking at habitability from galactic to microbial scales, so very broad range of scales. And I'm Tilman Spohn, and I'm your host today at ISI in Bern. This is the first time this year that I'm in Bern, actually, thanks to COVID. It kept me in Berlin for quite some time. We have the pleasure today, to have Ricardo Amils speak to us about life in extreme environments, and I will introduce him in a minute. But before I do, I wanted just to tell you that if you want to stay abreast of the seminar and other ISI activities, consider subscribing to our newsletter. You can do that at the bottom of any of the ISI web pages. With that, I turn to introducing Ricardo, who is an emeritus professor at the Department of Molecular Biology of the Universidad Autónoma de Madrid in Spain. He got his PhD from the University of Buenos Aires in Argentina, and held various positions around the world, ending, I understand, in Madrid. And here at this world-famous Center for Astrobiology. Ricardo is a world-known expert on extremophiles, and we thought that introducing us to the fascinating world of the extremophiles would be something that Ricardo could very well do for us. And I'm looking very much forward to what you have to say to us today, Ricardo. And before I hand the floor to you, let me just remind you that after Ricardo's talk, we'll have a chance for asking questions. And there are two venues of doing that. You can write your question in the chat, in which case I will read it to Ricardo and the rest of the audience. And Ricardo then will answer, or you raise your hand using the raise your hand feature on Zoom, and then I will switch between the two and ask Willie then to open the mic for you if you want to ask a question for yourself. And with that, I stop and hand over to Ricardo. The floor is yours. Thank you very much for speaking to us today.
Okay, well, I would like to start thanking the organizer for thinking that extremophiles is an interesting subject for the habitability discussions that you will have all along the next months. And just to go ahead with it, I mean, the program that I would like to talk to you today is, first of all, some historical notes, because I think it's important to understand how extremophiles evolve, the concept of extremophiles evolve. Also, we will go through the list of extremophiles that we know these days and their possible astrobiological interest. I choose to get deeper in the discussion in one new extreme environment that we don't know much about it, and it was predicted by Charles Darwin, which is the life in the deep subsurface. And I think that this will be very important in the future habitability discussions. And finally, I will go through other extreme environments that might be of interest to astrobiology. So historically, I think what is important is that the interest in extremophiles started in the 70s when Carl Woese described with his collaborators the concept, the new idea of Archaea. And to do that, he used extremophiles. I mean, I suppose that he did it, that's my point of view, because he had in mind that Archaea were related to the origin of life, and the scenario of origin of life should be extreme. But I really don't know, and I don't think he will ever tell us that, because he's not with us anymore. In any case, I think it's important in this context to mention that the first bibliographic reference on extremophile is due to Charles Darwin. Charles Darwin visited in his trip to South America a salt pond in which salt is obtained by evaporation, and he observed the presence of Artemia. And from that observation, he wrote in his journal in August 1833, almost 200 years ago, that if there is life on brine, in these brines, should be life in the subsurface, should be life associated to volcanic activity, to temperature. It will be life in the deep ocean. It should be life in the atmosphere and also in the perpetual snow. He predicted most of the extreme environments that we've been working in the last year to prove that he was right. But the first reference to extremophiles, or the interest of extremophiles, is an indirect one. We know that 2,000 years ago in China, there is a writing saying that there is a correlation between the efficiency of salt obtention, the efficiency of the production of salt in salt ponds, and the pigment color of these salt ponds. I mean,
Ricardo,
yeah.
Excuse me, but your slides do not advance. You have to advance your slides. Go to the next slide, or do you want to? You're speaking to your title slide all the time.
Sorry, I don't understand.
We just see your title slide all the time, and not next slide and so forth. So you have to advance your slides.
Okay. Is that right? So now?
No, we just see life in extreme environments, Ricardo Amils. You have to click on the next slide then, program and go forward.
No, but you don't see the slide changing? I'm sorry about that.
We just see the same slide all the time.
All the time?
Yeah. So far, we only see your title slide.
Jesus Christ. Well, and then we have a serious problem. I really don't know how to overcome this problem. I mean...
yeah, now we see salt ponds on Santa Pola.
Now you see it?
Yeah, we see a slide titled Salt Ponds on Santa Pola.
Okay. So now now you see a change?
Yeah, we see Extremophiles history too.
Okay, well then, sorry about that. I mean, I really don't understand what's going on. So probably we missed something before. This
is Charles Darwin. Okay.
The note of Charles Darwin in his journal in August 1883, advising the concept of the idea that the extremophiles should exist, and this has been proven later on that was right. And as I mentioned before, the first mention to the importance of extremophiles is a biotechnological one, which is almost 2,000 years ago. In China, they know that in the salt ponds, the presence of pigments, they increase the efficiency of the production of salt. And that you can see, I mean, I really don't know in which part of China this observation was made, but you can see in Santa Pola that in Spain, that the salt producers, they know that if halophilic Archaea grow, the efficiency, the productivity of the salt will be much higher. But the first microbiological reference to extremophiles was a problem in fish preservation in salt, which produced a very serious economic and social problem in the north of Europe, because in these conditions, halophiles, they grow and they can produce not only color, but only they can produce others from that putrefaction. And I think it's interesting in that sense to underline that the research in extremophiles is intimately related with food preservation. Think a system to preserve food and then you will find an extreme environment like high or low salt temperature, salt, acidic pH, sugars, low water activity, radiation. The problem of codfish preservation was solved by substituting the salt from ocean evaporation to salt from a mine. In that case, the probably the origin is the same, but the viable halophilic microorganisms in salt from mines is much lower. In the 40s, it was discovered an interesting microorganism, Thiobacillus ferrooxidans, that it was able, it was isolated from a coal mine and is able to produce acid mine drainage. So in other words, can grow using metal sulfides as a source of energy. And this gives rise to a biotechnology that today is very useful for producing metals. It's called biomining. And what we have here is the biohydrometallurgy, in other words, a new methodology to extract metals using bacteria or Archaea. In the 70s, everybody knows that Thomas Brock and his students, he isolated the first hyperthermophile Thermus aquaticus, very well known these days by the PCR. I think thanks to the virus, everybody knows these days what the PCR is all about. And but it took to Thomas Brock many years to publish his observations, because from the knowledge of the time, it was considered that at a very high temperature, life was impossible. In the 80s, after the description of Archaea, what happens is that it starts a time of competition to try to find the most interesting extremophile. So we find microbes that resist high doses of radiation, microbes that they were able to grow deep in the ice, in the poles, or in the big salt pond of Uni, or in the submarine hydrothermalism. I mean, associated to volcanic activity. To do that, we need a submarine like the Alvin to discover this part. And these days, probably the most interesting extremophile is in the LOL, in Danakil Depression. The only big problem is that right now is in a war zone, so it's impossible to go there to take samples. In the 90s, everybody knows that is the birth of astrobiology, and we know the important role of extremophiles in the roadmap of NASA and ESA in relation to the extreme environments. And also this opened up the interest of this subject in astrobiology, in the Mars exploration and the exploration of other possible places where life can develop. And here I like to show a picture of the Center of Astrobiology, which I'm very proud of it, being a member of it, because to the best of my knowledge, is the only center, the only place in which all the different subjects related to astrobiology are in practice. So it's very easy to meet an astrophysicist and discuss the latest knowledge on his area. In the 90s, and I think that's very important for the talk that we will have later on, it was the first time that the last frontier of Darwin's prediction, it was solved. It was to demonstrate that was life underground in the deep surface. In the beginning, all the data, they come out from artesian wells or mines, but in 2004 was the first paper describing life in hard rock, just drilling, getting rocks and extracting or demonstrating that life was down there. I think the latest discoveries that I think is interesting to mention is the discovery of CRISPR in halophilic microorganism in Santa Pola. As probably you know, this opened up the gene addition that today is very useful in biomedicine. And for me, it's very interesting also the discovery of ultra small bacteria, also known as PCR. That's, as you can see, they cover a very big part of the phylogenetic tree these days. Also, you can see that using this type of approach, we can these days know that Archaea, that eukaryotes, we come from Archaea. So we know that there is a group of Archaea called Asgard, from which we can see many properties that they share with eukaryotes. So that's probably the origin of eukaryotes. And I think talking about extremophiles is important to remember that most of the extreme environments are geophysical constraints. So that's conditions that are exist in a planet, and life is able to adapt to it. But I think also it's important to differentiate between tolerance and between real extremophiles. I know that this is a difficult concept, but I think it's very important because real extremophiles need extreme conditions to grow, while tolerance is only related to be able to solve the problems related with that temperature. And now I will show you a list of extremophiles just by alphabetical order. Just I don't want to avoid. I want to avoid political problems of saying my extreme environment is better than yours. And as you can see here in red, it's marked the ones that I think they are very important for astrobiology. Also, together to that, you can see the different extreme conditions in which these microbes they live, and in many of them you will find the champion. I mean, the record man this time at the condition in which it can grow. And you can see that we can find microorganisms at low pH, at high pH, inside of the rocks, at saturated solutions of salt, in very high concentrations of metal, with very low energy and nutrient source, also with a high, facing higher osmolarity, sugars, very high pressure, very low temperature, at high radiation dose, and high temperature, low water activity, absence of oxygen, the chaotropicity associated to salts that are very chaotropic, capnophiles, which are able to grow at very high concentration of CO2, etc. Until we arrive to the polyextremophiles, that today it's probably the best way to approach extremophiles, because normally extremophiles they are not facing only one extreme condition, they are sharing different extreme conditions, and that's the reason why we have to pay attention to different adaptation, different solutions to solve the problem of being exposed to different extreme conditions. And I thought that for this talk, it would be interesting to select one specific extreme environment to just get insight. And I thought that life in the deep subsurface will be very related to the discussions on habitability, that we will have in the next sessions. And in order to do that, I have to explain why we became interested in life in the deep subsurface. I mean, we know these days that there is some cumulolithoautotrophic bacteria. This means bacteria that are able to obtain energy from minerals that are able to produce sulfuric acid. And we are very lucky in Spain that we have Rio Tinto. Rio Tinto is, as you will see, an acidic river, pH 2.3, almost constant due to the buffer capacity of ferric iron, almost 100 kilometers long, with a high concentration of iron, but also many other metals. We always make the joke that you can find the periodic table in solution in Rio Tinto, and also you have a high concentration of sulfate, because iron and sulfate, they are produced by the solubilization, by the oxidation of pyrite. Rio Tinto rises in the heart of the Iberian Pirate Belt, and I don't think that I have to convince you that it's, you have a river with this characteristic thanks to the existence of the Iberian Pirate Belt, which is the highest concentration of metal sulfides that they are known on our planet. And I would like to make a very long story short. We've been devoting more than 30 years studying the microbiology of that river, and these days we understand quite well what is, which are the microorganisms and how the iron and the sulfur cycle, that they are the most important biogeochemical cycles in the water column, operate. And also lately we pay attention to the sediments, to the anoxic sediments. But thanks to being in the center of astrobiology, I've been able to interact very deeply with geologists to try to understand the substrates of these microorganisms and the products of these microorganisms, that they are geological. You can see in this slide, in the upper panel, terraces, sedimentary rocks that are being produced as we speak. In the middle part, we can see sedimentary rocks that has been dated 5,000 years ago, which is the oldest age that we know from mining activity in the Iberian Pirate Belt. In the lower part, we find, or we can see terraces that are older, and in a few seconds, you will see what is their date. Also associated to these sedimentary rocks, we have secondary minerals, like in the upper panel, we can see the famous jarosite. Well, jarosite became famous because it was found on it was detected on Mars. I mean, before that, only biominers, we knew about this weird iron and potassium sulfate. In the middle part, you can see spermanite, goethite, or in the last part, hematite. So these are different iron minerals that you can see along the river, in this case, produced by the biological activity. And the dated, when we date the oldest iron oxide deposit, we call it terraces from the river, we come out with a big surprise that they were more than 8 million years ago. This means that there is a lot of zeros in between the date of the oldest terraces in Rio Tinto and the first mining activity. So the idea that Rio Tinto was the product of mining activity, the contamination of mining activity, it doesn't hold anymore. So we should try to find another origin for the river. And one idea that we entertained was the idea of an underground reactor. I mean, we know that in the Iberian Pirate Belt, you have the substrate for these microbes. You need water to just facilitate the mass transfer reactions. And the microbes, you know, they are they like to grow in the places where they have a chance to obtain energy and cover. And to do that, we were lucky enough to be granted by a good ERC project in which the referees acknowledged that that was a very difficult project to develop. But as you will see, well, I think the end products of this project, I think they are very interesting, especially in relation to life in the subsurface. This is the place where the drilling took place, and you can see in the panel C the most important tool for geomicrobiology, which is a drilling rig to just obtain cores like the one you have in the displayed in D to generate samples to do deeper analysis. And that's the place, and it shows how we obtain the samples from inside of the cores with a Black & Decker modified to work at, and we extract the samples until we reach 45 degrees to avoid denaturation of nucleic acids. And it's in anoxic conditions because we know that underground there is no oxygen, and also in the sterile conditions to avoid contamination. which is the problem of subsurface microbiology. And here you see a display of the lithography, which I don't pretend that we go through it, but as you will see nearby, the display of the pirate content in these samples, and you can see that there is a lot of massive pirate associated to the drilling site that which we work. And from previous drilling projects done in collaboration with NASA, we learned that it's very important to have an ion chromatographer on site, because what is very important is to decide which are the best samples to do a deeper analysis. If you do that later on, it turns out that you maybe are analyzing things that are not that interesting. And here you can see a display of something that it really still, we don't have a good explanation for it. It's a nice distribution of nitrate all along the column. There is no doubt that this is so. We don't know the origin of the nitrate. There is a big discussion on it. I would like to prove someday that it is produced by microorganisms, but still is on hold. And associated to that, you see the nitrate. And here is not shown the presence of ammonia, but the presence of these three anions. I think what is important is it's giving us some insight that the nitrogen cycle might be very important in the deep subsurface. Also associated to that, you see a stochastic display of phosphate, which is a good news for microorganisms to synthesize their nucleotides, and also what you see, the distribution of sulfate, which is very stochastic also, and that's the product of alteration of the pyrite and other metal sulfides. And also using the same technique, you can see the presence of organic acid. This is a smoking gun that really proves that there is a lot of biological activity under our feet. And between all the organic acids that have been detected, I would like to underline acetate and formate, because I think they are very important for the discussion we are going to have later on. Also, we measure the gases occluded in the different samples that we obtain. And you see, again, good news for microbes, because you see a lot of hydrogen, the best electron donor, and CO2, good carbon source for autotrophic organisms. Also, we measure total protein content and sugars. And again, these are all different methodologies that are trying to show that under our feet, there is a lot of biological activity. And being in the center of astrobiology, we are lucky enough that we can use the methodologies that they are working on it in order to fly someday to Mars and try to see signs of life. And this is what you see here on display. This is called the LD300 chip. It's a chip based on 300 antibodies with different specificity. And in the bottom line, in the C display, what you can see is the signal along the different levels of the drilling of different types of bacteria that have been identified using this technology. Also, we use next generation sequencing, and here you see the display of the diversity along all the column using Illumina. And here I will show you a detail of this data, because they are much easier to see. And you can see some bacteria that they are able to obtain energy using hydrogen, and others that they can oxidize iron using nitrate. They are iron oxidizers, nitro reducers. And also we use enrichment cultures. These enrichment cultures is a fast way to know which kind of activities are active in the underground. And you can see that there is microbes that are able to solubilize pyrite. This was expected. Also iron and sulfur. And also there were iron reducers and sulfate reducers. Altogether, this shows that the iron cycle and the sulfur cycle are important in this specific ecosystem. We also prove the presence of methanogens using different substrates, and also methanotrophs. I mean, in this case, there were consortia or Archaea and sulfate reducer bacteria that are able to use the methane produced by the methanogens. And also we look for the nitrifiers because there is a lot of nitrate in the system, and acetogenic bacteria in order to explain the high amount of acetic acid that we were detecting in different samples. Also, from these enrichment cultures, you can isolate microbes. And if you can isolate these microbes, we isolate 27 microbes from different samples, from different enrichment cultures, sorry. And some of them, and you have a list here, we sequence their genomes. So in other words, we know the gene possibilities, I mean, the genes that this genome has in order to explain the different metabolic activities that we detect. The technique that I like the most is in situ hybridization. I always say that I'm like Saint Thomas. I like to see things because most of these techniques that you just have the display, the number display, and you have to believe that that's so. But in the case of in situ hybridization, if you have the appropriate probes, you can detect the microbes and only detect them, but you can see how abundant they are. And here in a sample obtained at 407 meters, you can see the fluorescence display or the fluorescence of the microbes against the probe, again, using the beta proteobacteria probe or the gamma proteobacteria probe. This is in the upper part. In the lower part, you see the positive hybridization reaction or signal using a specific probe against acidity-bacillus ferrooxidans. And here is a general display of different types of activities in general that we detect. I don't want to go into all the details, but in the upper part, you can see the presence of Archaea, also the presence of biofilms in which you can find bacteria and Archaea together, probably collaborating in a metabolic way. Underneath in the panel E and F, you can find acidobacteria. In G and H, you can see chloroflexi. In E and I and J, you can see anamoxis. This is a very interesting reaction. It's the first time that has been detected underground. In the next panel, you can see the activity of sulfur-reducing bacteria, which they were expected. And the lower panel, you can see Tesarococcus, which is iron oxidizer nitro-reducing bacteria that we are very interested in it, and also a rhizobium, which probably this is a surprise because the question is what a rhizobium is doing at 497 meters below surface. To the best of our knowledge, there is no plants at this depth. Anyway, and here you can see the display of the lithography, the pirate content, the different elements that we detect, the gases that we detect, the metabolisms we detect using enrichment cultures, and nearby you see the positive signals doing in situ hybridization. And I think the lesson to take home from this the resume of all the data that I show you is that there is many different metabolic activities, but we don't see, as it happens in the sediments of the ocean, a gradient, so that the farther you go, there is a less diversity of microbes. In this case, you can see the diversity is maintained all along the 620 meters display that we studied. And one of the critical points in the geomicrobiology of the deep subsurface is that normally, first of all, there is very few data. But in addition of the few data, the problem is that normally authors, they use only one technique to describe the diversity. And usually this technique is next generation sequencing, which is okay. But from our point of view, it will be nicer if instead of using only one technique to identify diversity, use different ones. And the methodologies that they are based on different technologies, so that they are not using the same approach. And this is what you see in this display. This is the list of the most important genera that we accept or we believe that they exist in the subsurface of the Iberian Pirate Belt, because it has been detected by more than 2 different technologies. I mean, underground in blue, you see the technologies that we use, but as you will see by the density of the colors that there is some bacteria that they appear in using many more different approaches, different methodologies all along the column. I will underline the importance of acidophilic, an iron oxidizer in anaerobic conditions, Pseudomonas, that is very versatile, sulfobacillus, a sulfur reducer, or Orthocerococcus, again, another iron oxidizer. And if now we try to put all this together, we would like to display a model of how we see the interaction, the interrelationship between the different biogeochemical cycles that we detect underground. You will see in the left, in blue, the nitrogen cycle, in red, the iron cycle, in green, the sulfur cycle, and the carbon and hydrogen cycle are in the far right. And you will see here the display of the name of different microbes that they have been identified at different depths, and in red, the ones that we sequence the genome. So in other words, we know the genes that are able to maintain the reaction in which we associate the microbe. And also in the display, you will see the different electron donors and electron acceptors that we detected using different methodologies all along the column. And what this is trying to give you is the image or the impression that there is an important collaborative work underground. So you don't find different micro niches in which one microbe is isolated and doing its work, but I think there is a very important interconnection between the different biogeochemical cycles, which I think that will be very important to maintain the ecosystem alive. We always claim that if there is one very smart microbe, all the system will collapse, because the cooperativity between the different biogeochemical cycles will not maintain. And this is the lessons we learn in this project. So we have detected a high level of functional diversity. The most important biogeochemical cycles have been identified. Of those, the nitrogen cycle, we believe that is more important than what we thought before we started this work. Most of the activities in the nitrogen cycle have been detected underground. Also, we detect, and this was very important for us, bacteria that they are specialized in oxidizing iron in anoxic conditions. And we believe that from this data, we can conclude that we can explain the origin of the extreme conditions that we detect in Rio Tinto. And what we have to ask is, was it the interest of an environment like that? And of course, I must confess that in this field, there has been an incredible advance due to economical reasons. This is the base of biomining, and many companies, they invest a lot of money to try to understand how the mining of the future will be done. But obviously, we are astrobiologists, and our main interest was an astrobiological one. And now I will show you few data obtained from different space missions, which are related to what we just showed. Well, the first one, and was very important for us, was when the MER opportunity showed using the Mossbauer spectrum that jarosite was on the surface of Mars. Jarosite is a very peculiar mineral that can be only produced in acidic conditions. Also, other the analysis of other samples show the presence of goethite and hematite. But for us, jarosite was very important, and that's what I would like it to underline. Also, the data of the Mars Express showing with a radar that there is, without any doubt, liquid water in the underground of Mars. Also, we must remember that there is a big debate on the methane that has been observed by different, and that's very important, by different methodologies on the surface of Mars. And today I read a paper that just appeared saying that now they can explain why the data of the Curiosity are different from the ones from the ESA orbiter that claimed that it didn't detect anything. And the Curiosity did an interesting experiment. It just heated some samples, and they obtained different types of volatiles. That it's interestingly enough, in addition of telling us the kind of minerals from which these volatiles come from, I mean, they are good elements that they are very useful for the microbes that we find underground. So I think we have to pay attention to this kind of minerals in order to explain how microbes can live in the subsurface. I think it's very obvious. I can just show the presence of CO2, the presence of oxidized sulfur and reduced sulfur, which claims for a putative sulfur cycle, and the best electron donor that we know in microbiology, which is hydrogen. And here what I do is to compare the properties that have been detected in Meridiani Planum, or have been reported from Meridiani Planum, and the correspondent properties of that we detect in the Rio Tinto surface or the Rio Tinto subsurface. And you can see from the first part that there is no difference between the content of the iron minerals that have been detected in both ecosystems. So that's the reason why these days Rio Tinto is considered a good geochemical and mineralogical terrestrial analog of Mars. Also, I point out that we should pay attention to the soluble salts that have been detected on the surface of Mars, because if someday these soluble salts were in solution, any putative life had to resolve the problems of osmotic pressure. In other words, to be halophiles. The temperature, we know that the surface of Mars is very cold, but we know that underground, the temperature that there is a gradient of temperature, a very different temperature of what you see in the surface. So we need to know what is the temperature gradient that exists on Mars before we can predict, or we can make other predictions. Methane. Well, there is claims that methane has been identified on Mars. There is no doubt that methane is being produced in the subsurface of Rio Tinto. And just to conclude, we know that there is life in Rio Tinto. Obviously, on Mars, we have to prove it. But I am absolutely sure that in the very near future, when we will have samples from Mars on the Earth and we will be able to analyze them carefully, well, we will be able to solve this question mark. And just to finish, the actual conditions on the surface of Mars, intense radiation, very oxidant conditions, low temperature, doesn't seem the ideal place for life to develop. If I go to Mars, I would like to have a shovel just to make a hole, you know, and try to sneak in underneath, because I think life in the subsurface will be much more possible than on the surface of Mars. And we claim that we would like to have a specific missions, specific Mars mission, just to drill and to try to find out data on the gradients and the possible biosignatures that might exist on the underground on Mars. And also, well, we can claim that the use of extreme environments in order to are important if we want to understand the properties of the microbes that we might find on Mars or in another bodies in the solar system or out of the solar system. And also, it's important to know that these extreme environments are good tests to test the instruments that are going to fly in next missions in the future. And of course, astrobiology is not finishing on Mars. I mean, we know that there is other places in the solar system that we would like to explore. And here you will see a display of putative possible terrestrial stream analogs to collect information, to get prepared for this kind of missions. One is Tires Lagoon in Spain that has, it's here because it has a mineral content very similar to what has been reported from Europa. Also for exploring Europa, it will be very important to know how life is distributed along the ice column that we have in the Antarctic. Also, in Salar de Uyuni, it's a place in which temperature gradient is very important. Also, halophiles are there, and there is also the presence of very chaotropic salts, which they have been detected in some other bodies out of the Earth. In the Antarctic, we can see that there is microbes that are able to oxidize iron from metal sulfides, and they are being disposed in the surface. You can see the structure of sbermanite. This is the kind of mineral that we see in the surface of the Antarctic. I mentioned Dalol. Dalol is a very interesting model system for the way it was Mars in the time that big volcanic activity existed. Also, Islandia is a good place for studying many interesting subjects related to extremophiles, or the Atacama Desert, which is also a good ground to study possibility of life in oligotrophic conditions. But we shouldn't stop in the solar system, because we know that the number of exoplanets that are appearing every day is increasing. And with the new instruments that we just put on orbit, I know that the number of extrasolar planets will start to increase in an incredible number. And here it just showed to remind us that in one star, the TRAPPIST-1, in which the presence of different exoplanets has been studied, there is two of them, the exoplanet E and F, that they have the properties, they have the characteristics to claim that it's habitable, sorry about that. It can be habitable because water in the surface will be maintained. And I don't think any more things to do but to acknowledge my colleagues and collaborators from the Center of Astrobiology, that they help us, or they've been the protagonists of getting most of the information I just showed you in the subsurface of the Iberian Pirate Belt, and many students and different institutions that have been collaborating with us in the last 35 years to try to understand what Rio Tinto is all about and what is its interest in astrobiology. And I thank you very much for your attention.
Thank you very much, Ricardo, for this fascinating introduction into the world of extremophiles. So I'm sure that Ricardo, is ready to answer questions. And as I said in the beginning, you have two ways of posing questions. You can write your question in the chat, and I will read it to Ricardo, or you raise your hand using the raise your hand tool of Zoom, and then we'll give you the microphone. So I'm looking at the chat. There is a thank you, but I don't see a question yet. So maybe I ask a question. So if you compare, what you did, Rio Tinto to Mars, and you said that it would be important to know the subsurface temperature. Well, we have, with InSight, we have measured not the temperature very deep, but the thermal conductivity. And then we extrapolate the temperature to the range that you were addressing, a few degrees, then we would probably have to go to tens of meters. Is that something you would be happy with, or would you suggest maybe they're at lesser depths because they would tolerate lower temperatures on Mars than they do on the Earth?
Well, it's very obvious that you don't need 37 degrees to grow. I mean, obviously, sequeophiles are showing us the possibility to grow at much lower temperatures. Obviously, they are much lower than the kind of life that we detect on the surface. But I point that this in the talk because I think it's very important to understand that the temperature in the surface is not related whatsoever with whatever you have underground. And we are talking about 600 meters, you are right. But I mean, the gradient of temperature that we detect in the Iberian Pirate Belt, it's only 10 degrees in 600 meters. But I will claim that at this very moment that we are speaking, the radars are trying to locate liquid water, not at that depth in Mars, and I hope that we will find it, or else we will have to wait for astronauts to go to Mars and be able to drink in a conventional way to go that deep.
Yeah. Okay, there's a question here in the chat. So how is your take on the Mars methane mystery? Sources, causes of variation in space and time? And Rudy asked that question.
No, but could you repeat, please? Because
so how is your take on the Mars methane mystery?
Yeah.
What does the source?
Oh, okay. Well, as probably, well, I'm an investigator of the Mars Express, so I was there the first time that methane was detected on Mars. Unfortunately, this spectrometer is not functional anymore. But the methane was detected by independent methodologies with radio telescope from Earth, and also the Curiosity have detected. The problem was that the Curiosity was able to see it. It didn't see it again, but there is no doubt about that. I mean, I talked to the colleagues that they have been involved in in the analysis of this data, and the problem was that the orbiter, the ESA orbiter, didn't detect it in the similar position. But I think 1 week ago it was a paper published trying to explain how this can be possible. That different approaches that disagree. And my personal interpretation is that I don't have any doubt that methane is being produced. We don't know if it's biological or geological. That's why it's very important that Curiosity trap some methane and really looks for the isotopic fractionation. But yet we will have to wait until more data are coming out to try to really tell us the end of the story. But I don't have any doubt that there is methane on the surface of Mars.
Are you familiar with the Copahu volcanic system in Argentina that is very similar to Rio Tinto, but volcanic in origin? River with jarosite, shredmanite, hematite, etc.? It's a question by Johan Barekamp.
Yeah, I'm very familiar because I did my PhD in Argentina, and I have good friends that they did this work. The only big difference between the volcanic origin of this type of minerals is the size. I mean, this is a lake inside of a volcanic activity. Rio Tinto has a very peculiar distinction. It's a 100 kilometers river. You don't see any mineralogy, neither different microbiology from any acidic mine in the world. So we are not claiming that they are unique in the terms of the microbiology or the mineralogy. What is unique is the size. I mean, you should imagine how much bioleaching you have to produce in the Iberian Pirate Belt to maintain 100 kilometers river pH 2.3 due to the concentration of ferrigate.
Oh, Candy Jessup asks, I am wondering if you can comment on how important water is to the Tinto underground microbes. You speak of hydrogen receptors. What is the source of the hydrogen? CH4, water, something else?
Well, I must confess that we still don't understand many things. because the project is still working on. Most of the information that we got from the well I show you is, I didn't show it because there was too much information. I didn't want to bother you. But the rocks, they are seeing neutral pH. What I think it's producing the acidity in the river is the presence of ferric iron, because when ferric iron hydrolyzes, it's precipitating ferric hydroxide as liberating products. So what you need to understand, I mean, in order to explain the generation of Rio Tinto, is how you start to generate ferric iron. Ferric iron that, if it's produced in anoxic conditions, will also, like in the aerobic conditions, oxidize metal sulfides, dissolve metal sulfides, and will increase the amount of iron and the sulfur associated to it. But again, to prove that, you need obviously to, I mean, we don't have the flow data in order to explain all these things. I mean, the project was driven to try to show that there is a lot of diversity down there, and this diversity can maintain metabolic reactions that might explain what you detect in the subsurface.
Okay, actually, I don't see, oh, there is a thank you, but I don't see any question in the chat, and I don't see any raised hands. Maybe I can come back to Mars for a little bit. You know, what about, you know, saws, you know, reducing the melting temperature or percolate as a fluid? Can you comment on this perhaps a little bit? Do we need water, or can we have other fluids, you know, that would, you know, allow us a biosphere?
Yeah, well, I mean, these days we know that life on the surface is not possible due to the extreme conditions. No, I know, but I mean, if it's underground, I think we need to know the origin of this water. Remember that there was a lot of water on Mars, but we still don't know what happens to it. I mean, some people claim that is now a part of it is underground, other parts were just splashed and they are flying around the universe. But you don't need a lot of water. That's something that maybe I was not able to communicate. Underground, inside of a rock, there is no much place for water. You know, I mean, it is well understood that you don't need a 10 kilometers ocean, deep ocean, to just have life. I think you need water for the reactions of matter transfer. And I think we don't know much about what happens in the pores of the rocks. I think that's an interesting subject that we need to pay attention to it, because there is researchers that claim that the origin of life maybe didn't need membranes because they were using the pores. I mean, the pores as the containers for the metabolic reactions that were happening. You know, I think the subsurface life, I mean, from my point of view, this is something new. We don't have enough information. We need to work harder in order to collect them, because I think it's an important asset if we want to find life elsewhere in the universe.
Well, we have any indication, you know, from the InSight mission that the first meters or so on Mars, at least where the InSight landed, is very porous. You know, it's a very porous regolith. So, you know. Okay, anyways, so thank you very much, Ricardo, again. And thank you very much to those in the audience. We will close this session in a minute. Just let me point out that next week, David Catling will speak about the habitable zone, a critical appraisal. And you're all invited, you know, back to his talk at the same time, you know, same location. Looking forward to seeing you again. And again, thank you very much, Ricardo. Have a good evening. And to those of you in the US, have a good day. Okay, bye-bye.
Ciao.